Good morning, everyone. I'm Atif Iqbal, M2 student of Photonics Engineering at UGM. And today I'm going to present my internship project, which I did at LPCIM, Ecole Polytechnique, within Horao project. So let's talk about the brain cancer. The glioblastoma is an aggressive brain type of brain tumor, and the key risks in this type of tumor cancers is that the incomplete resection during the surgery. Why? Because when we detect this type of tumors, we use MRI and other technologies, but they help us to detect only before the operation. But during the surgery, the neurosurgeons showed us this open brain of an human and asked us, where is the tumor? Can you answer it? It is impossible to distinguish between a healthy brain cell and a tumor. So, but fortunately, the brain, human brain is made up of fiber tracts, which are an anisotropic medium with which we can do the polarimetry and we can help the surgeons. Like if we detect our sending stroke vector and detecting the outgoing stroke vector of our light from the sample, we can by applying the mathematics, we can have this type of images of the brain. And now I can clearly say to the surgeon that where is the tumor. It is very clear because the tumors are not an anisotropic medium and they are very distinguished from the healthy brain cells. So how we can detect this type of images? To detect this type of images, we create a prototype within a Horao project, and with this prototype we send light and using the different polarizations, and then detect it with with the DOFP super pixel cameras, the all polarization states, and then we apply the mathematics and have the images of the sample. But the problem with this prototype is that we can only detect at single wavelength, like we cannot scan the whole spectrum of the light with this type of prototype because the cameras we were using were only operating at single wavelengths. So to solve so like why we need to scan the whole spectrum of the light, why it is necessary. Here is an normal human brain's sample, which is under two different illuminations, like at 550 and 650. And in this green circle, you can see clearly a difference between the sample we at the different wavelength. We have like a lot of information which is hidden. So we have to see the sample at multiple wavelength to clearly judge the sample. Why this happens? Because there's a lot of blood in the human body, in each part of the human body, and which absorb the light at different wavelengths, and it causes the artifacts in the sample images. So to solve this, we applied some upgradations in the prototype, like we characterized our liquid crystal based polarization state generator and worked on different parts of the prototype. So the major change in the whole prototype was to introduce an new kind of detection system, which like the previous system was using an super pixel system, which can detect like the different polarization states at 1 pixel. But in an RGB camera, there's a new addition of an Bayer filter inside this super pixel, which can allow us to detect the same information at different wavelengths as well, at different wavebands as well. So with this, the prototype now can detect the sample at from 4 channels to 12 channels. Like now we have the information at least at 3 different wavelengths at same time. And with this, the neurosurgeon can have like more information from the sample. And then to implement this new upgrade, we worked on the calibration and control part of the prototype to operate the system at and also we upgraded the illumination source of sample. And with these upgradations, we become more near to this type of images, which can be achieved at real time speed of at the video real time speed of acquisition. And that's all from my side. Thank you.
Hello. Okay, thank you for this presentation. Is there any question? Okay, let's try to find something interesting to ask. Remember, DSC student, you can get half a point to ask question. No? Okay. So, yes.
Thank you for working. What is the resolution of the your imaging system?
For us, the resolution was not the matter, because it is the wide field imaging. So like we were using the camera which have like 5 micropixel resolution, which is not like a lot of high resolution. But for the neurosurgeon, it was like to have more real time imaging. So for us, like the resolution was not a target.
Okay. Thank you.
Is there any question here? So I was wondering, what is the gap between your prototype and a real...
The prototype with the single wavelength is like ready to be commercialized, because it is a project of Swiss national. It is a project of Switzerland government, and they are going to implement in their hospitals, starting from the intellectual Bern. And the multispectral prototype can be implemented in 1 or maybe 2 years.
Okay. Okay, thank you. So.